Project M.A.G. DNA Analysis Report Analysis Date, the 24th of September 1988. Lead Geneticist, Doc. Linus Ovku. Subject Information. Species, Drosophila melanogaster, fruit flies. Identification Code, DMMS #12. Exposure Type, Preliminary Testing Phase, Mutagenic Compound. Summary of Genetic Changes. The DNA analysis of the mutated subjects exposed to the mutagenic compound in the preliminary testing phase of Project M.A.G. reveals significant alterations in various genetic markers. The observed changes are inconsistent across multiple subjects, indicating a pattern of uncontrolled mutagenic influence on the subject's genetic makeup. 1. Mutations in regulatory sequences. Observation. Prominent mutations have been identified in the non-coding regions of the genome, particularly in regulatory sequences responsible for controlling gene expression. Potential implications. Altered regulatory elements may contribute to the observed changes in phenotype, including variations in behavior, growth patterns, and reproductive cycles. However, with the knowledge we currently have, we have no way to distinguish whether or not the mutations are controllable and/or predictable. 2. Chromosomal rearrangements. Observation. Structural changes in chromosomes, including translocations and inversions, have been identified in the mutated subjects. A rare case of mutation is that the chromosomal makeup changes to a type we have never seen before, which allows the creatures to reproduce on their own without needing to mate with another type. Potential implications. Chromosomal rearrangements may lead to disruptions in gene linkage, potentially influencing the subject's overall physiology and development in ways we do not know how to control yet. 3. Gene expression patterns. Observation. Marked changes in gene expression patterns have been noted in mutated subjects compared to control subjects. Potential implications. Altered gene expression may contribute to observed phenotypic changes, such as variations in pigmentation, morphology, and reproductive behavior. In extremely rare cases, we have also identified a rapid change in size to strength ratios which are beneficial to our goal. 4. Presence of novel genetic elements. Observation. Analysis reveals the presence of previously unidentified genetic elements in mutated subjects. We have yet to identify the origins of these genetic elements and what they are made of, but are aware that thus far they do not show any signs of threatening the life form. Potential implications. The introduction of novel genetic elements suggests potential integration of external genetic material, possibly influenced by the mutagenic compound. This has resulted in certain experiments being able to break free or even merge with their holding cells in studied chambers. This is alarming to us, but also adds a new interesting spectrum to our research. We continue to delve deeper into our research to uncover the truth behind these novel genetic elements. 5. Epigenetic modifications. Observation. Epigenetic modifications, including changes in DNA methylation patterns, have been observed in mutated subjects. Potential implications. Altered epigenetic regulation may play a role in shaping the subject's response to environmental stimuli and their adaptive capabilities. We have seen this particular part play a role in diets, adaptability to humidity, temperature, and physical mutations to adhere to the environment. Conclusion. The DNA analysis of mutated subjects exposed to the mutagenic compound in the preliminary testing phase indicates a complex interplay of genetic changes, including mutations in regulatory sequences, chromosomal rearrangements, altered gene expression patterns, the presence of novel genetic elements, and epigenetic modifications. These findings provide valuable insights into the transformative effects of the mutagen on the subject's genetic makeup. Further investigation and correlation with observed phenotypic changes will be crucial for a comprehensive understanding of the molecular mechanisms underlying the mutagenic influence. Additionally, the identified genetic alterations will inform subsequent phases of Project M.A.G. as we progress toward achieving the project's objectives.